Well, hello, I'm Dr. Rushmore, and I'm happy to welcome you to a video that I'm calling Fun with Connective Tissue Cells, Mast Cells versus Eosinophils. Now, over the next couple of weeks, you're going to be taking a gander at the different types of cells that live in connective tissue, and you'll be asked to identify them on the basis of their morphology. For most of the cells, it's fairly straightforward, but for these two cells, for the mast cells and for the eosinophils, it's a bit trickier. And so the goal of this video is to help you suss out that difference. The first thing, of course, is to identify and to be able to identify what eosinophilic granules are. Both of these cells contain them, and it's largely on the basis of this sort of cellular component that you'll be making your identification. And linked to that, it's really important to be able to tell when you're in connective tissue. And so what we'll first do is take a look at how defining connective tissue spaces can lead to two very different ideas on what a cell may be. And then we'll give it a shot, mast cell versus eosinophil. So to start off with, we will go to this section. This is the ileum and the Peyer's patches. Here's the serosa here. Here's the peritoneal cavity, the lumen of the ileum. This is muscle here. This is connective tissue spaces here. This is an epithelial sheet, which is corrugated and has invaginations. And underlying that is the connective tissue space, the lamina propria layer, that we'll be taking a gander at today to look at these cells. So, goal number one is to figure out what eosinophilic granules look like. And once we figure that out, then we can identify which of these cells we're talking about. So if we go into higher magnification, around 40, we can see that these little small circular profiles, much smaller than the nucleus, pop out at us. And we see them very nicely demonstrated here. So there's some there, around here, and here. And so since this is underneath the epithelial sheet, we're down here, epithelial sheet is here, we are going to be tempted to make our identification there. Oops, pardon me for a second. There we go. But here's where we have to consider things a bit more carefully. If I were to draw the basement membrane of this epithelial sheet, there we go. It would be something like this. And we would follow it around a little bit to get a better understanding of what it looks like. And one thing that we note as we follow it along is that the lumen here disappears as we sort of take a section that's glancing through the edge of the epithelium as it dives in. And we see that here the cells are somewhat darker. And over here, as we go into the sheet towards the larger connective tissue spaces, these cells are somewhat darker. And so when we look again at our area here that contained our granules, first of all, it looks very similar to these cells and these cells, but it is not connected, as far as we can tell, to the lumen. Well, actually it is. We just don't see the connection. So for instance, if I was to take a look at this, and here's the lumen right there that connects eventually here, and if I were to take a section like this, what it would show is a rounded section like this, and the basement membrane here shown in green. And so these things right here, these little circular profiles, are finger-like invaginations of the epithelial sheet. And here's the lumen here that, for instance, may connect out of the plane of section with the lumen there. And so these profiles are actually part of the epithelium, but we just don't see the connection. So why is this important? Well, it's important because if we want to identify whether a cell is a mast cell or an eosinophil, we have to consider the fact that both of those cells are in connective tissue. And we have to control against the possibility that what we're looking at is not actually connective tissue. And so these cells, in fact, what are known as Paneth cells, and we'll learn about those later in the course. They have eosinophilic granules that they discharge, the contents of which discharge into the lumen and then go into the lumen of the ileum. And so our first goal really is to make sure that in our identification we're not looking at the epithelial sheet itself, because they have cells with eosinophilic granules. Okay, so now we have that under our belt. Let's go back to our little drawing pad and just take a look at what the difference between the cells should be. Alright, so we've got two cells. I said we have two cells. There we go. Eosinophils, or Eos, as you might see them, and mast cells. Okay, so these guys we will find in the connective tissue. The mast cells are slightly bigger than the eosinophils, but it's really difficult to tell on the level of the light microscope when we're trying to compare them. The mast cells are characterized first by a nice round nucleus with a rim of heterochromatin within the actual nuclear envelope and a nice basophilic nucleolus. The nucleus of the eosinophil is lobed. It can have 2 to 3 lobes, and it looks something like this. And each of these lobes is connected by a string of nuclear membrane, and it's easy to think about these as sort of sausages connected by the sausage linking stuff. Alright, so this is what we have, and for the nucleus, but they also both contain eosinophil granules. Now, this is why the eosinophil is called the eosinophil, and these granules may change a bit in color depending on the stain, but they should always be pinkish. The mast cell granules in the section that we're going to look at is highly eosinophilic, and in fact the granules are indistinguishable from the eosinophilic granules. But in other stains, for instance with toluidine blue, these granules can be purplish blue. And so it's really on the H&E stain that we're interested in titrating these cells from these cells. Okay, so overall we have two main features: a lobe nucleus versus a central round nucleus, and granules. So since the granules are the same for both cells, it's really this guy that's going to be important in telling the two apart. Alright, so let's take a gander at the section itself, and we'll go through a couple of examples. And we'll come back here and consider a couple of practical issues. Okay, so what we're going to do now is take a look around, take a little tour around, and see if we can see a mast cell. This is going to be the easiest cell to see. And what we're going to be doing is looking for granules just like the granules that we saw in the epithelial sheet. Just same color as this. This is epithelial sheet, except in the connective tissue. And so here's some granules. And here is a round nucleus. So this one is fairly straightforward. I think there's no problem here. This is a mast cell. If we go up here in the same field, we can see one round central nucleus. This is actually a nice example, and sort of the pink granules that we associate with eosinophilia. Now notice though that the granules are a little less distinct here, and this is probably a fixation artifact. And sometimes, the optimal times, you will identify granules that are distinct and nice and pretty, but sometimes you're going to identify the color, because fixation artifacts or staining artifacts will tend to sort of merge these things together and make them clump. And so that's something that we definitely have to consider. Alright, so we have two mast cells, and so let's see if we can track down an eosinophil. So what we're looking for in an eosinophil is a cell with multiple lobes. And, well, once we know what multiple lobes are, then we are in a better position to make that distinction. And so another cell that has multiple lobes, fairly numerous, is a cell right here. and this would be a neutrophil. And here's one lobe, and here's another lobe. So reasonably small, heterochromatic, and within the same cell. And we see one here too. There's 1, 2, 3 lobes. This is a neutrophil. Now an eosinophil is very, very similar to this. It has a lobe nucleus, anywhere from 2 to 3 lobes, but it also has the pink eosinophilic granules. So we'll see what we can do here, and there's probably one here. So there's a lobe, and maybe even this is a lobe. And here's that eosinophilia, which is characteristic of the granules that we have seen earlier. Okay, we'll keep looking around a bit and see what we can see. And when we do, we find a couple of other things. We find something along these lines. See this guy right here? This is definitely eosinophilic. Same granule quality as we see in the epithelial invaginations, but we don't see a nucleus here. So this is going to be something that we'll consider in a second. The other thing that I want to consider is, let me see if I can find an example up here, is kind of a tricky example as well. And let's see, I thought I saw one a little earlier. Give me 30 seconds. Okay, something along these lines. Okay, so those are the two instances that we're going to sort of think about. This is certainly eosinophilic granules. There is a round nucleus here, but both of these things present some problems. So this is really where things get tricky. So we had, oh dear Lord, we had two things to consider. Sorry about this. We have one cell, which we had something like this, plasma membrane, and then granules within that. We did not see a nucleus. And so the question is, what is this? Is this the mast cell, or is this a eosinophil? Well, let me remind you of what we're talking about here. So mast cell, oh dear Lord, dink. There, mast cell has a round nucleus like this, with eosinophilic granules. And I apologize, this is a terrible looking mast cell. It kind of looks like an eye. And then the eosinophil has lobed nuclei, and the lobes are usually a little more heterochromatic, but sometimes they're not. And of course this has lots of granules. Again, not a very good drawing, so forgive me. So what could this be? Could it be a mast cell? Well, the answer to that is yes. And the reason it could be a mast cell is it could cut through here. So we don't actually see a nucleus, but we see a cut through part of the cell. Could it be an eosinophil? And the answer here is also yes. Making a cut through here would allow us to see something like that. So answering this question doesn't really have an answer. It could be both, and therefore we can't identify it. And there are going to be instances where you can identify things, whether that's from a practical nature, that is the plane of section is a problem, or whether it's from a staining issue and you just can't really figure it out. I can assure you that any examples we may give you in sort of a test environment are going to be very, very clear. But if you're just sort of interested in figuring this stuff out for yourself, understand that sometimes it's not worth making the distinction, because sometimes you can't have a good answer. With that in mind, let's take a look at the second profile that we're interested in. And this was slightly bigger, if you'll recall. It had a nucleus here, sort of centrally placed and round, and then it had granules. So the granules and the fact that it was in the connective tissue space told us unequivocally that this was a mast cell or an eosinophil. Okay. Now we have a round nucleus, and so could this be a mast cell? Mast cell is round nucleus, it has granules. This is round nucleus, this has granules. And so the answer to that would be yes. Okay, easy cut through, let's say, here. So we get cleave off a little bit, but not the entire center of the nucleus. We would get something like this. All right, the harder question here, and the one that's going to blow your mind, is could this be an eosinophil? And the answer here again is yes. So something like this or something like this would also give us a profile of that. Somewhat smaller. And here it's fairly heterochromatic, but that could be because we're hitting the rim of the nuclear envelope or because we're going through an eosinophil. So in this case, it could be either. So the best thing that you can do if you want to identify this versus this is to identify the eosinophil in all of its glory. That is, identify the multiple nuclei. In this case, the best answer, since we can't see multiple nuclei, we can't assume that something is there. I would call this a mast cell. But with reservations, because it's not a perfect mast cell. It's smaller, and it could be an eosinophil. But the likelihood, or what I'm going to sort of put down, is that it's more likely it's going to be a mast cell than it's an eosinophil. All right, based on the fact that what we have said a mast cell is going to be is a round nucleus with eosinophil granules. So I guess one of the take-home messages here, apart from being able to identify this, is that the problems in identifying a mast cell versus an eosinophil come with problems in interpretation. There's always a gray area in interpretation, and understanding sort of the limits of what you can say in the context of the stain and the preparation and the morphology of the cell is really where it comes down to in terms of identification. So practice, good luck, and have fun. Bye-bye.